as many of your readers and viewers know, small cell lung cancer is one of the most aggressive subtypes of lung cancer. And it usually presents as metastatic disease upon initial diagnosis. So we were interested in trying to characterize the tumor diversity and the immune microenvironment and see if these factors may explain the high metastatic potential of small cell lung cancer. And so to do this, we profiled small cell lung cancer patient tumors from both primary and metastatic sites at single cell resolution using single cell RNA sequencing and proteomic spatial imaging using the MIBI platform. And what we found was that small cell lung cancer has a very high tumor diversity that far exceeds the other common lung cancer subtype, lung adenocarcinoma. And a lot of that tumor diversity was driven by canonical transcriptional subtypes, the most common of which are subtypes A and N, which each stand for the transcription factors ASCL1 and NeuroD1, both of which have different therapeutic vulnerabilities. And we found that in contrast to the classical neuroendocrine subtype A, the variant subtype N is found at increased frequency in metastatic sites. And along with this pattern, it adopts a neuronal looking phenotype that engages this gene program called axonogenesis, which is where the cell actually creates these projections, these axonal-like projections similar to neurons. And it's interesting because previous studies have also shown that axonogenesis can actually help facilitate metastasis in small cell lung cancer. But beyond this tumor diversity, we were also interested to see if there were any universal features of small cell lung cancer that could also help explain metastasis. And so what we found was that there exists a cancer subpopulation that is recurrent and is marked by high expression of this gene called PLCG2. And we found that it's present in a large proportion of patients regardless of the predominant small cell lung cancer subtype. So PLCG2 is interesting because it's known to mediate cell motility. And we confirmed in both in vitro and in vivo functional studies that PLCG2 overexpression actually increases metastasis in small cell lung cancer. And it also, and taking away that expression also ablates that effect. So we also found that the presence of this PLCG2 high cancer population predicts much worse overall survival. So the other aspect of our study focused not on the tumor cells themselves necessarily, but on the immune microenvironment, which is important because small cell lung cancer is known as a relatively immune cold disease, with the addition of immunotherapy having only marginal benefit to overall survival. And so we confirmed that small cell lung cancer does indeed have a higher rate of immune cold tumors. But even with the tumors that had relatively immune warm features, those immune cells were largely sequestered from the main bulk of the tumor. We also found that, we were interested to see if any changes in the immune microenvironment could relate to tumor diversity itself. And we found that the variant subtype N tumors do have a decreased abundance of immune cells. And within that immune microenvironment, the T cells actually showed a greater dysfunction with an increase in inhibitory Tregs and a decrease in CD8 positive effector T cells. We were also interested in looking at the myeloid compartment, and we found that the small cell lung cancer in general has these myeloid cells that actually resemble pro-fibrotic macrophages that are found in a completely different type of a benign lung disease called idiopathic pulmonary fibrosis. And these pro-fibrotic myeloid cells are actually correlated with the presence of the PLCG2 high recurrent tumor subpopulation that I was talking about before. And so it's interesting to speculate whether these pro-fibrotic myeloid cells might be depositing the substrate in the environment that's needed to allow for these PLCG2 high tumor cells to metastasize. And so we made all of this data publicly available on the Human Tumor Atlas Network website. So you can go there and you can easily visualize and explore all of this data and also download it for your own independent analysis as well. And so I think you were also asking whether what the potential clinical implications are for all of this. And I think this provides a mechanistic picture of how small cell lung cancer, and particularly both canonical and subtypes like the variant subtype N and other non-canonical novel populations that are high in PLCG2, how these different subpopulations of small cell lung cancer might metastasize, and how the immune cells that surround the small cell lung cancer might be helping accelerate this process.